Good afternoon and a warm welcome to the third in our series of webinars we are running to take the theory of marginal gains, making small incremental improvements in a process to give an overall significant improvement, and applying this to the world of IVF. Speaking in the webinar today, we're delighted to have Dr. Dave Morrell join us. Dave has worked as a clinical embryologist since 1986, training in Manchester, where he also completed his PhD studies. He has managed several laboratories in the UK, including being responsible for the designs of labs in Leeds and Daresbury. He joined Origio as Director of Embryology in November 2011, becoming Director of Clinical Support for CooperSurgical in January 2019. Dave previously served as chair of the UK Association of Clinical Embryologists and Association of Biomedical Andrologists, as well as being involved with several working groups, notably the HFEA Expert Group on Multiple Births After IVF. In his current role, Dr. Morrell is part of CooperSurgical's medical affairs team, providing technical and scientific support, as well as education and training. He has specific responsibility for lab designs, clinical trials, and lab outputs. We're also very pleased to have joining us today Dr. Steve Troop. With nearly 35 years' experience in the field and a PhD in male infertility, Steve has risen through the ranks to have been the scientific director of Liverpool Women's Hospital's Hewitt Fertility Centres and more recently IVI's UK centres. As well as his day-to-day operational, managerial and research responsibilities, Steve has often provided advice to regulatory bodies, educators, the commercial sector and colleagues around the world. He's particularly proud to have been both chair and president of the Association of Clinical Embryologists and a founder member of the newly formed Association of Reproductive and Clinical Scientists. Steve now works independently as a consultant reproductive scientist based in the UK. So it's normally just the three of us for Marginal Gains webinars, but we have a very special guest today. A big welcome to our colleague, Dr. Steve Levitt. Steve has worked in embryology in the public and private sector since the early 90s in the UK, Canada, Nigeria, Sri Lanka and Kuwait. Steve joined Wallace in 2006 and became Global Business Director in 2010 and worked as National Sales Manager for Ferring before joining Life Global as VP International Sales. Since 2018, Steve has worked as Director of Clinical Applications at CooperSurgical Fertility Solutions, supporting IVF clinics in optimizing culture systems and introducing new products and protocols, including today's subject of embryo culture. We have a few international delegates today, which is wonderful. You're most very welcome to join us from wherever you may be based around the world. We and our panel members are based in the UK and Ireland, so any references we do make to regulation will have the UK and Ireland in mind. If you have any questions at any point, please enter them into the chat box. We love your questions, so please ask away. We have 30 minutes today and there will be time at the end for the questions. Thank you very much for your attention. And Dr. Troop, I'm going to hand over to you to get things going. Thank you.
Great, thanks very much, Emily. And good afternoon, everybody. Thanks for asking me to take part in this webinar. Just before we get into the detail, I'd like to start just by quickly reminding you what marginal gains are all about. The principle of marginal gains was really highlighted around 10 years ago by a guy called Sir Dave Brailsford, who was the performance director for British Cycling. And as you can see from his quote, marginal gains is essentially the notion of improving every single component of a system by a small amount. And although those individual improvements may themselves not be noticeable, when they're all added together, they then become, they lead to a tangible and measurable improvement. And this can be represented graphically on the slide. And I think many of you, I'm sure, will be aware of the unprecedented success of the British cycling team in recent years, much of which is attributed to the marginal gains approach. So if I could have the next slide up, please. So what we're trying to achieve in this series of webinars is to apply this marginal gains approach to the IVF lab. And clearly there's an awful lot to think about here. This article, which many of you will know, by Don Rieger, suggests that there might be some 175 laboratory factors that could influence outcome. And in this particular webinar, as Emily mentioned earlier, we're going to concentrate on optimizing culture conditions. And I guess this is particularly a particularly important aspect of the lab, as it affects all of the gametes and embryos that pass through your lab. But before handing over to Steve, let's just take a quick look at the poll, which I have the results of on my screen now. And I think you can see them as well, I hope. So the first question, how often do you test pH? There is an interesting range of answers there. Nobody is testing pH more frequently than monthly, which is interesting. Certainly I've been into labs where they test pH on a daily basis, but the majority of people there are not testing pH. And I'm sure Steve will have something to say about that. I guess the people who don't know whether they're testing pH or not are probably not testing pH either. The majority of people, moving on to question 2, seem to be using mineral oil, which again is perhaps not unsurprising. And in terms of whether oil is washed, whether you use pre-washed oil, or whether you wash the oil in the lab yourselves, the majority of people there, as you can see, are buying pre-washed oil. And again, I think Steve will comment on that. So I'll hand over now to Dr. Levitt to take you through some of the details. Thanks very much.
Just unmute myself so you can hear what I have to say. Thank you, Steve, and welcome everybody. It's always a great pleasure to speak to people on the embryology world, so I look forward to sharing some thoughts with you. As Steve has pointed out in the graph by Don Rieger, there are many factors that go into the success of an IVF clinic, and specifically to an embryology laboratory. And we haven't got time today to go into all of those factors, so we're going to focus on some of the culture conditions, particularly pH, osmolality and oil. But we have to bear in mind that those factors can only be optimized if the rest of the system is truly optimized as well. So we mustn't forget about the environment, the air quality in the lab, the equipment that we use, and the validation of that. And then, of course, in terms of outcome, that we get good quality embryos, we can select the right ones for embryo transfer, and that we measure our performance, which is critical, with some suitable KPIs. And there are other resources to look at those. So bear in mind, this is only a snapshot of what we can do. There's much more to it than those factors we're going to discuss today. So I put this slide on to demonstrate the change really that's happened over time, and why some of the factors we'll talk about today have become so critical in optimizing our systems. So for many years, we used a very simple IVF culture media, and then thankfully commercial media became available, and we started to use that, and over time that has been developed. And so there were two schools of thought that quickly developed: those that use sequential media, that provided a media for the early cleavage stage development, and then a blastocyst development media, which obviously had to be changed on day 3, and then sort of hand in hand, but was slow to gain popularity, was single-step media. So Life Global developed single-step media in the early 2000s, and then over time, and particularly with the use of time-lapse, single-step media has become more and more popular. But in those early days of using single-step media, it was also changed on day 3. So we refreshed the media on day 3, but obviously replaced it with the same media that the embryos had already been cultured in. But more and more common nowadays is this use of single-step media that's non-renewed on day 3. So we basically make our dishes the day before we're going to use them. We put our embryos in there, and they're going to have to stay in that environment for 5, 6, or potentially even 7 days. So the strain and stress that's put onto those embryos in culture and the culture setup has become more and more important in the last few years. And we're going to cover why why I think we should measure some of these parameters, or at least be cognizant of them, in terms of optimizing our system going forward. And the first one I'm going to cover is pH. And it was interesting to see that the majority of people don't test pH. Historically, that's been the case within embryology, partly because it's been very difficult to find a suitable methodology to give accurate and reproducible and results that you can trust to act upon. But the reason we should be looking at it, of course, is that in the human embryo development, the human embryos initially follow the external pH in the culture media, so they are affected by their surroundings, and fluctuations in that external pH can affect an embryo's development. The embryos themselves do possess mechanisms for regulating their internal pH. We'll talk in a little bit about some of the particular stages that we need to look at and be extremely careful of. But the modulation of internal pH is an energy requiring process. So the less stress we can put on the embryos to modulate that pH, the better they're going to be in terms of health. And it's been demonstrated in hamster and other species that these slight changes in pH, if they last over any length of time, can result in structural problems and developmental issues. Although as the embryos develop, it looks as if they are less susceptible to these changes. So by the time they get to the blastocyst stage, they are able to modulate their internal pH to a much better degree. So pH can intimately affect the infrastructure within, or the internal structures within the embryos themselves. But there are two areas where we need to be particularly careful with pH and pH control, and that is when you denude eggs, when the cumulus cells are removed, then the eggs themselves lack the ability to modulate pH. So any fluctuations in the outside environment is going to significantly affect them. And again, as with in the hamster model, these pH changes seem to affect the internal structures, the cytoskeletal elements within the egg, and can cause problems with future development and division. So certainly when you denude eggs, when you do ICSI, whenever you're handling a denuded egg before it's been fertilized, and we need to be extremely careful to make sure we maintain a very stable pH in the optimal range. So how do we do that? Of course, I'm sure most of you are well aware that this is a dynamic process that is the link between the concentration of bicarbonate in the media and the concentration or the partial pressure of CO2 in the incubator itself. For most commercially available media, the range that's given that's suitable for use in human embryology is somewhere between pH 7.2 and 7.4. This is actually quite a vast range, and certainly within each laboratory, we should work to determine within that range what is the optimal pH for our embryo development. And as you all know with your incubators, we set the pH by modulating the CO2 concentration. So the bicarbonate concentration in the media is set by the manufacturer. So wherever you buy that from, the concentration of bicarbonate is set. And unless we do something to the dishes that we prepare them, so we either dilute the media some way, potentially with protein supplementation, or we allow the media to evaporate, then we can get changes in bicarbonate concentration. But if the bicarbonate concentration remains static, then changing or manipulating the CO2 partial pressure will change the pH. And if we can keep CO2 concentration stable and bicarbonate concentration stable, then we will maintain a stable pH. So getting our dish preparation, we'll talk about this in a little while, is extremely important, so that the concentration not only of bicarbonate, but of course all of the other components of the media itself remain constant and don't change over time with evaporation. So dish preparation is going to be a focus. When we're talking about setting up our dishes, we need to be aware of several factors that are going to play a part in that setup. And it's fairly well established now that the equilibration time for dishes is much longer than we first anticipated. So in the early days, certainly when I was working in the early 90s, we used an open culture system in humidified incubators, but over time we've changed to using drops under oil. and dependent on the volumes that you use and the type of oil, then it's suggested by Steele and Conahan here in 2008, and there are other papers now that say that you need a minimum of 10 hours to equilibrate your dishes correctly. So we really need to bear this in mind, and certainly to make dishes on an emergency basis is going to be very problematic. Better to make more dishes the day before that may be in excess, rather than trying to make dishes on the day, which is going to be problematic in terms of equilibration. So one of the questions we're often asked is, what CO2 concentration should I use for the media that I'm using in my lab? And unfortunately, there are too many variables to really be able to do that from a distance. We really have to be in the lab to test to make sure that all of the systems are set correctly, and the CO2 concentration that we're using gives us the target pH. So altitude is a specific issue, not so much so in the UK, but in other parts of the world. For example, in South America, there are clinics that use single step media, and they have to use somewhere in the region of 11 or 12% CO2 to get the correct pH of the media. And this is, of course, due to partial pressure of the gases, and as you increase in altitude, the partial pressure decreases, and therefore you need a higher concentration to maintain the correct pH. But whether you supplement the media yourself or whether you buy pre-supplemented media, the volumes and times that you use for incubation and equilibration will also play a part. And one of the major concerns with embryologists is, how do we measure pH in a suitable way that will give us accurate data that we can act upon? And unfortunately, in the embryology world, pH meters are a significant hindrance to getting that correct data. We often find that calibration solutions are not up to date, they're not made properly. pH meters often don't like protein, they're susceptible to change in temperatures, and certainly don't like oil in close proximity to the probe. So pH meters generally are not suitable to measure a pH reading in a media that's under oil, and certainly closer to what the embryo might see in situ. And so I and others now have adopted the use of blood gas analyzers. These allow us to do a much more accurate reading, collecting the sample from under oil in a very quick and easy way, and giving accurate, reliable and reproducible results that we can act upon. So there now is a methodology that we can use which can help us to maintain a stable pH in our culture system. Of course, if we're not going to use a bicarbonate buffered system where we're manipulating eggs or embryos outside of the incubator, we may want to use what's called a Zwitter ionic buffer. And the most common used in the UK and elsewhere are MOPs and HEPs. And you can see from this table, they vary a little bit in their pH buffering range, and also in their pKa, which is an indication of the relative acidity of the buffer itself. And as you can see at 37 degrees, the pKa here gives an indication of the middle of the buffering range that occurs at that temperature. And you can see some differences here. And both will work perfectly fine outside of the incubator, and are really preferable to using a bicarbonate buffered system, which will off gas fairly quickly and potentially give us big changes in pH, which could be problematic. And so just highlighted HEPs here. Just wanted to point out that one of the issues that often is related to me is that HEPs is toxic to human embryos. And this is a little bit of a misnomer because it's the fact that bicarbonate's not present that the embryos won't develop fully in HEPs, but actually the HEPs buffered media itself is not toxic and will allow maturation, fertilization and embryo development, as you can see here. So the use of HEPs shouldn't be a concern, and we should use it in an appropriate situation where we're outside of the incubator. Other things that can affect pH, of course, are volumes, and particularly volumes of the media and the oil. And this is a nice demonstration on this picture here of a small change that was made that can have a profound effect on the culture conditions. So if you look at the two cells on the left here, they're made up with the media plus 35 microliters of oil in these cells, and on the right just an increase to 50 microliters. And the ones on the right are blown up to show you that visually you really can't see any difference between the two, whereas even at this lower magnification you can see significant difference between these two cells as they were set up on the right and after 7 days on the left. And then if we look at the table above, you can see that there is a difference in the final pH between the two setups with just a 15 microliter increase in oil. The most disturbing factor though here is the change in osmolality. And so almost 100 milliosmol per kilogram change over the 7-day period. And this would take IVF culture media into the toxic range for our embryos. So bear that in mind at the back of your mind when we progress a little bit about volumes, both of the media and of the oil. And so things to consider when we're looking at pH and how we use our media. So whether we use pre-supplemented or we supplement media ourselves, and from a regulatory point of view, we should always use pre-supplemented media. It's CE marked and is controlled by the manufacturer, so more reliable and less variable. And you can see some of the other factors here, and so the how often you open the incubator, and if we had time, there's some data to show you how that can impact on outcome, and some of the other factors that you might expect. Pay close attention here to humidity. In the early days, again, for the first many years of embryology, we mainly use humidified incubators, but the trend now is to use dry incubators, and these pose a specific question that we need to answer in terms of stability of our culture, and we'll cover that in a second. So one of the final factors that I'm always asked about is an embryo transfer. So we go out of our way to protect the environment throughout the embryo's development, and we come to embryo transfer, and this is often the place where the lab take least care about the control of pH. And often I've been into labs and done testing where the culture conditions are okay, but the dishes that are used for embryo transfer are often open without oil and are left for too long without any cover, and the pH can change quite significantly. So a use of an oil overlay can help prevent that. So the use of oil itself, if the tip of the catheter is washed prior to taking it through to the embryo transfer procedure, is no issue. It is possible to use Heapes buffered media without problems. And I just made a note here that as well as denuded eggs, the other area we need to be extremely careful about is when we warm vitrified embryos, because they, again, for a short time, 3 to 4 hours after they've been warmed, lose their ability to regulate their internal pH. So we need to be very careful that we maintain a stable pH once we've warmed our embryos. And of course, we need to be cognizant of temperature, as we do with everything in the embryology world, to make sure that that's at an optimal setting. So we're going to move on to one of the more current debates, and that's around osmolality. So pH is a dynamic process, and we can change the pH in the media with changing the CO2 concentration. But the problem with osmolality is once it starts to rise, it is impossible to get it back down to where we would want it to be. So it's an increasing problem over time. And you can see from this study, it's in the mouse, but you can see that as osmolality increases, certainly above 300 milliosmoles, then we start to see significant impact on the development of the embryos in the culture system. And the use of non-humidified incubators, the use of small drops, and insufficient oil can really exacerbate this problem. So this is just this is a study done by Jason Swain, looking at how the making of dishes can affect the change in osmolality. And you can see from this graph that there are some factors that can have a significant impact on osmolality right from the very beginning. And as I say, over time, it can only get worse. So if you've already introduced a rise, this is going to make the situation worse as you go on. So the temperature that you make the dishes has an impact, so we should always try to use room temperature. How you introduce the drops into the dishes, so use an underlay or an overlay method, and we can discuss that. This is mitigated in some degree by using dishes that have preformed wells, and certainly a significant factor is the volume of the drops that you use. And as you decrease the volume, you increase the surface area to volume ratio, and therefore you increase the risk of evaporation and therefore increase in osmolality. So when we're using small drops, we need to be extremely cautious because evaporation can happen very quickly and certainly we should minimize airflow where possible, but not have any external air conditioning vents, etc., pointing towards our workstation where we're making dishes. Of course, we generally have the workstations or with the airflow on to maintain sterility. So are some significant factors here that we need to take into account when we're making dishes, and it seems incredibly trivial. And any embryologist that's been in the field for a while will sort of question why we should be paying attention to this, but because of the change in our culture habits, the dishes become extremely important in maintaining a stable environment. And this is a slide that my colleague Dave Morrill produced, which shows quite nicely that the surface area is of significant importance when we're looking at evaporation and the potential change in osmolality. So as you can see here, the note at the bottom, the rate of change of osmolality is influenced by the surface area. So the volume and how you make the droplets is going to have an impact on that particular issue. And so the use of preformed wells is going to minimize surface area to volume ratio, and therefore going to help you with a stable culture condition. And there are many dishes with preformed wells available now, and certainly should be considered for use in the embryology lab. So to demonstrate here, this is about the volume of oil. So as we saw with those cells that were made up to check pH, you also see a change in osmolality. And so oil becomes a very significant factor. Not only the type of oil that you use, but the volume is of particular importance. So if you can see on the graph here that as you increase the volume of the oil, the change in osmolality is decreased. So with just a change from 8, 10 or 12 milliliters of oil, or 15, you can see that going from the blue to the light blue, there's a significant difference between the change in osmolality over time. And so volume of oil is important. And the rule of thumb generally is if you look at your dish and you look at the peak of the drop, we should have at least a 2 millimeter clearance of oil above the top of the drop to give us the best protection that we can. And so this was an internal experiment done within CooperSurgical, which basically shows exactly the same thing. And you can see that some osmolality changes can happen fairly quickly. So this is a change between 4.5 mils and 6 mils of oil overlay. And after 48 hours, you can see there's a very large increase in the osmolality of the media. And this is now above 300 milliosmoles, which is potentially going to be detrimental to embryo development. So don't be tempted to go down to very small drop sizes within your culture setup. 20 microliters, I would assert, is the minimum that you should use. And if you can use more than that, that will be helpful. And certainly don't minimize the volume of oil that you use. It will help you maintain that stable culture system. There was a question on the poll about the use of mineral or paraffin oil. These, of course, are related to the US, which always has to be slightly different from Europe. So in the US, it's called mineral oil. In Europe, it's called paraffin oil, but they're exactly the same things. They come from the same extract from the petroleum industry, and they have these long chains of hydrocarbons, and the length of the chain of hydrocarbons dictates the viscosity of the oil itself. So they're exactly the same. They're same formulation, whether you use mineral or paraffin oil. And then just as two final notes about oil, there's a lot of press at the moment about whether we should use heavy or light oil. Does the density make a difference? And you can see from this graph here that as time goes on, there seems to be a benefit, if you look at the middle column here, to using a heavier or more viscous oil. Certainly seems to give more protection as time goes on. So if we're going through uninterrupted culture for 5, 6, or maybe 7 days, plus remember the day before when we've made the dishes, so these could potentially stretch to 8 days, then it becomes increasingly beneficial to use a heavy oil to protect your culture system. And then the final point here is really to look at whether we should use washed or unwashed oil. So this is a study done by Dean Morbeck, where he looked at unwashed oil, oil washed with just media plus protein, or the water source that was used to make the media in the first place. And you can see that certainly washing the oil with either water or media plus protein is beneficial in terms of blastocyst formation. And therefore we should certainly consider using pre-washed oil in our culture system, because it may help remove these lipophilic and hydrophilic endotoxins that may be present in the oil. So again, looking at how the oil is tested, using the highest quality oil that you can use in your laboratory will minimize any risks from the oil itself. Storing it in the cool and outside, away from sunlight or direct light, will also help. So just to summarize the use of oil in IVF culture, it's incredibly important, but often ignored as just something we add to our dishes. We don't think about the type of oil, the volumes of oil that we use, etc. But of course, it helps with minimizing pH shift. It significantly helps with changes in osmolality over time. Will act as a temperature sump, so reduces the risk of minimizing temperature fluctuations. And of course, it's also a physical barrier. So it does protect our dishes and our drops from outside contamination, be it particles, bacteria, etc. And of particular note in our laboratory that we want to avoid volatile organic compounds. So the mantra that we should work by is really use the maximum amount of oil that we can in our culture dishes to avoid oil sealing. So we don't want the lids of the dishes to stick. So we need to be cognizant of that. But certainly if you use a heavier oil, it actually prevents spillage as well. So that might be more easy to add more volume to your dishes without the risk of spillage. So pH, osmolality and oil are critical to optimizing our culture system, and we need to be looking at them all of the time and making sure that they are optimized for our particular systems. And testing in the lab is the only way to know what actually is going on, and ensuring that we maintain a stable environment throughout the culture time that our embryos are in the incubators. So that's it. I'm sorry it ran over a little bit, but hopefully it's given you some food for thought. And we're open to some questions if people have them. And if we don't get through them today, we can also answer them at a later date if you can send those through to us. So thank you for listening.
Thank you very much, Steve. Really enjoyed that. And as Steve said, we're open now to questions. We've had a few come in already, but please do keep them coming in. So we'll crack on with some questions then. So Steve T, my first one to you. If we choose to wash media, as Steve has mentioned about washing oil, in our lab, does that affect the CE marking?
Yeah, I think this is an important point. Steve demonstrated very clearly there that using washed oil is better than not. But I think one needs to avoid the temptation of washing oil in the lab yourself, because my understanding is that anything that you do that isn't described in the instructions for use for that product will effectively invalidate the CE marking for that product. So certainly in the UK, where we are required to use CE marked products if they're available, that could lead to a problem for you. So my recommendation will be to buy pre-washed oil.
Great. Okay. Sound advice. Thank you, Steve.
Just add to that, Emily.
Please do.
From looking at our product, the one oil that we have that isn't pre-washed actually states in the IFU that it can be washed. In fact, I think it's a recommendation. And whilst we know a lot of people don't wash it, it is in there, so you could do it, and it would be covered by the IFU.
Great, that's reassuring to know. Thank you. Next question. We'll pose this one to you then, Dave. How consistent is the concentration of bicarbonate in different batches of media?
Yeah, that's an interesting one, and I think what you'd have to say is that because as commercial manufacturers, most of the base media that we use will be produced in bulk. The control over individual components of the media is much better than it was, for example, when we were producing our own medium. So I guess that there'll always be a degree of variability between batches, but that is very much defined and controlled within the specifications of the product when it's registered. So part of the FDA approval or the CE marking, those limits of variability will be defined. So there'll be a small amount, but it'll be much better controlled than any sort of in-house preparation. And bear in mind, of course, that as part of the quality assurance and quality control, the key parameters are tested before the batches are released.
Brilliant, great, that's good to know. So a little bit within the acceptable defined parameters, as you say. Following on from that then, we'll go back to Steve T. Why test pH if bicarbonate is consistent?
That's an interesting question, and I think there are many labs, as we saw from the poll at the start, that aren't testing pH at all and are relying on the fact that the bicarbonate in the medium will be consistent and the CO2 level in the incubator will be consistent. But I might just throw this to Steve Levitt, because I think I recall seeing data that the CO2 level on the display on an incubator doesn't necessarily reflect the CO2 level that's inside the incubator itself. So the safest way to approach this is to test pH and not rely on those sort of surrogate markers for pH, if you like. I don't know what you think, Steve.
Yeah, and there was that's very true. And there was a very nice publication that was in the Embryologist, actually, I think in 2010, by Rusty Pull, who's an embryologist in Texas in the US. And he did some work in his laboratory looking at measuring CO2 levels, and so doing a control chart about where he expected CO2 levels to be. And hand in hand with that, he did pH measurements. And quite interestingly, sometimes when the CO2 varied, the pH didn't. So if you just measured CO2, you might intervene. If you had your control chart and it went above where you were expecting the CO to be, you might tweak it, or it may go below and again do the same thing. But actually the pH remained stable, and vice versa. So the pH may change in your media, but the CO2 wouldn't reflect that. So I think as well, if you take a single, whatever media it might be, and we run it in lab A in a similar incubator with similar CO2 concentrations, and we did exactly the same in lab B, you wouldn't get the same pH, because there are so many other variables around humidity, about how dishes are made, how they're equilibrated, etc. And so, like everything that we do, the only way to know what's actually going on is to test it and to look at it in your setup. And that's why frequency of testing becomes a question that many embryologists ask. So there are clinics that test with every new batch to take some of these variabilities into account. I mean, where the fact that bicarbonate may vary, it's actually by a very small amount. You know, you're not going to see vast amounts. You would see much more variability, for example, if you buy pre-mixed gas for your incubator, because the tolerance there is much greater than it is with culture media. So some people would test every time they change their CO2 cylinders, whether it be pre-mixed gas or supplied from laboratory. So again, you need to work that out in your lab, in your system, what you feel comfortable with to assure that your system is staying stable. Some get external people to come in every now and again. So there'll be labs that test on their own, and then they ask an external person to come in to validate what they're doing and to make sure that, you know, they're actually getting the right results. But since we've used a blood gas analyzer, the testing of pH has become much simpler and much more available to more labs, and it's becoming an increasing parameter that people use. And ultimately, embryos aren't interested in CO2 concentration. it matters not to them. It's only the effect it has on the pH in the media. And so that's the parameter we should measure.
Thank you, Steve. On a similar theme, we've had a question: how often should you realistically test pH? Which I think you've answered there in a.
Yeah, I mean,
it depends,
as I say, it really depends on your quality management system and what you're comfortable with, and really doing an assessment of where you think it may change in any aspect. So, of course, if you buy a new incubator, it makes sense to do some initial studies to look at pH stability. But then, as I say, you might want to do it on a regular basis that you're happy with, or with every new batch of media, or with CO2 changes, or whatever. I mean, it's really up to the individual labs to determine what they're comfortable with to ensure that they've got stability in their culture system.
Lovely, thank you. And on the theme, you mentioned the blood gas analyzer. So our audience are intrigued by the blood gas analyzer. So do you recommend one? And can it be used for? Oh, sorry, there's a couple of other questions as well. I'll get them all out.
Yeah.
So can you recommend one? Can it be used for all media, so MOPs and HEPs? And what kind of volumes of media do you need?
Yeah, all great questions. The two that we currently use in the IVF field are an epoch reader made by Siemens, and it's called an iStat made by Abbott. We tend to use the epoch system in Europe and Abbott in the US. Both we've done. Tim Schimmel, who works in the Center of Excellence in New Jersey, has done a lot of validation work on both of those systems, and looked at the relationship between what we can measure and the actual pH within the drops, and the validation has proved very positive, and you can really rely on the results. First question is very easy. You can't measure zwitterionic buffers in a blood gas analyzer because the control of temperature is not where it necessarily is, and the way that the machine works out the pH is slightly different. So as you know, HEPs and MOPs have to have a very controlled temperature to give the correct pH reading, and so the blood gas analyzer doesn't maintain the temperature in the right range to do that. And also, as I say, the way it works out pH is not a direct measurement, and it doesn't work with that type of buffer. The difference between epoch and iStat in terms of volumes, they both need about 100 microliters of media to do the test. We usually take a slightly larger sample than that, but you can do it in embryoscope slides, in other time-lapse slides as well. If you're very careful in how you sample, got to avoid getting air or oil into the sample, but otherwise it works fine. And just on that point, to reiterate, time-lapse incubators are not immune from osmolality changes. It's a nice paper published recently from Barcelona with Mestres et al. Showing that osmolality rises do occur in time-lapse incubators because they're dry generally, and the volume of oil that you use in the particular slides is also relatively low. So you do need to be cautious when you're setting up, even in that scenario. I think sometimes people think it's a protected environment, doesn't apply, but all of these things we've discussed today also apply in that scenario.
Thank you. Actually, we've had a question specifically saying how would you recommend to measure pH with the epoch in a time-lapse incubator such as the embryoscope? Do you have any tips for that?
Yeah, you can. You don't have to use the wells that are there. You can actually flood the bottom of the slide and put oil over the top and do it that way. The new embryoscope, the Embryoscope Plus, does have a slide you can buy for doing pH testing. They have a specific slide to do that. I suspect it was a little bit, I don't know, I shouldn't say that because I don't know for certain, but it wasn't available for Embryoscope. It is available for Embryoscope Plus. You can do it with the Embryoscope slides. You can do it going into individual wells, but you need to be extremely cautious. So if you just flood the bottom of the slide and cover it with oil, you can get enough volume to do the test quite happily.
Lovely. That's a great tip. Thank you. Another question here. There are some studies that still recommend to use humidity in culture. I would like to know, why should dry culture be chosen if we are increasing the evaporation rate? And then there's also a really nice comment. I'm just going to read this out. Thank you, Millen. It's great to learn from the experts. So thank you for your comment there. Dave, perhaps we go to you on that one.
Yeah, I'm sure Dave's got his views on dry versus humidified incubation.
They each, I mean, the simple answer is they each have their pros and cons, and we've tended to move towards dry systems because first of all, time lapse is technically easier with a dry system because you don't have issues around humidity and the cameras. But also in general incubators, just taking water reservoirs out avoids risks of waterborne infections and the likes. So there was a general assumption, I think, that with oil overlays, evaporation couldn't occur, and obviously we've determined that that isn't true anymore. But there are clearly pros and cons to each approach, and I think it's probably fair to say that, again, it varies from lab to lab to determine which system actually works best in their circumstances. I don't think there's a one size fits all. But essentially, the water reservoirs come with other risks that dry systems don't have. But you do have to tweak your systems, as Steve has described, to control osmolality, particularly with volumes of oil, I think.
Great, lovely. Thank you for that. How quickly will media change pH when left outside the incubator? Steve T, should we go to you for that one?
Very quickly. Again, it depends on the shape of the drop, depends on the volume of the drop. It depends on whether you, for example, have a funnel over the dish blowing CO2 down onto it. So again, there's loads of variables, but generally the answer is very, very quickly indeed. And steps need to be taken to minimise that, if at all possible. And as Steve Levitt suggested, using a HEPES or MOPS buffered media, if you are handling gametes or embryos in air, is a far better approach than to take the risk. But we are talking seconds or minutes and not hours for pH to change. It changes very quickly.
Got to be quick there then. One last question for Dave. Given pH and osmolality concerns, what is the ideal drop size? And I think Steve did reference in his presentation a minimum drop size of 20 microliters. But have you got any more thoughts on that?
Yeah, and I think that's an interesting one, because my perception is that we've tended to move over time to smaller and smaller drop sizes, and I think that the work that Steve referenced from Jason Swain's lab about the impact of drop size on osmolality changes at dish preparation means that certainly below 20 microliter, you have to be much more fastidious in the way you do your dish preparation to avoid those changes. So I think certainly above 20 microliters is a good starting point, and I think 50 and above perhaps is a more potentially more useful starting point for many systems. And I think, as Steve also referenced, it's not just the volume, but also the type of dish you're using. So weld dishes where the surface area is significantly reduced for the same volume would seem to have clear advantages.
Lovely, thank you. That's all our questions then. So we'll wrap things up. So it just remains for me to say thank you very much to Steve Levitt for presenting for us today. A very thorough presentation, very interesting. Thank you very much. And also to our regular panel members, Steve Troop and Dave Morrell, and also to Danielle, our Zoom master, who you can't see, but she's behind the scenes, keeping us all in line and keeping everything running smoothly. So thank you, Danielle. If there are any lab or theatre procedures or aspects of these procedures that you would like us to cover in particular during this Marginal Gains series, please do let us know. You can either contact your account manager or you can email us at customerserviceuk@coopersurgical.com.